study leaf senescence in Arabidopsis thaliana. Leaf senescence is characterized by the leaves turning from green to yellow as they age. It's what gives fall that characteristic color change. And during this, nitrogen and other macromolecules are broken down and recycled from the dying leaf and then mobilized towards the growing parts of the plant, such as the fruit and seeds. So these fruits and seeds are often eventually harvested for consumption in agriculture, so it's really important to understand this process. We think that improving the efficiency of leaf senescence could improve the nitrogen mobilization process, which would help farmers not have to use so much nitrogen fertilizer while they're feeding the plants. That would be better for the environment, and it would save money because it's expensive to make that nitrogen fertilizer. So I specifically study the relationship between flowering time, also called bolting, and leaf senescence. So during stress, such as drought or heat, plants can bolt early, and then when they bolt early, they also often senesce early. And this is bad because it can cause inefficient senescence in the plant. So I want to know, what is the molecular connection between early bolting and early senescence in Arabidopsis thaliana, which is our model plant organism that I studied. I hypothesize that there's gene expression changes that occur at the time of flowering that signal the onset of leaf senescence that cause it to occur and couple it to flowering time. So to study this, I had to design a mutant that would both flower early and senesce early. So previous literature showed that ATX mutants flower early, and then data from our lab showed that ATX mutants would likely also senesce early. And that is what I found after isolating this. So here, shown on the left, ATX mutants bolt about 5 or 6 days earlier than wild type, which doesn't sound like a lot, but when you realize that the lifespan of the plant is about 60 days, that's about 10% of its life just taken away right there. It also has early senescence, which I showed here by a significant decrease in leaf chlorophyll concentration in the mutants compared to wild type. And it also has significantly increased expression of 2 genes here that are known to have high expression during senescence. So all this together shows that these mutants both have significantly early flowering and significantly early senescence. So after that, I'm looking for flowering time specific gene expression changes that signal senescence. I can't look at one gene at a time, there's too much to do. So I use a technique called RNA-seq, and what that does is it looks at every single expressed gene at one given time in a given tissue, and then I can compare all these expressed genes across different samples. So I designed this time series experiment that looks at gene expression changes in these early flowering plants shown right here, those mutants that I showed you, and I compared those expression changes to the vegetative wild type of the same age. So it's important to note that they're the same age, but they're developmentally different. So with this, I was able to look at all the differentially expressed genes in the plants. I detected over 19,000 transcripts overall in each sample and then compared those computationally across each sample. This is an example of what that can look like. So here's one of my wild type samples at the first day. Blue shows low expression while yellow shows high expression, and you can see along the y-axis is 19,000 different genes and their expression levels. So I was able to compare this level of expression from this wild type sample to the mutant samples over time to see what's actually different. So first we found some really interesting hypothesis supportive gene expression changes, and I just want to remind you the hypothesis was that there's gene expression changes that occur at flowering time that signal senescence. And 3 of these gene expression changes were involved with nitrogen. 2 of them were involved in nitrogen metabolism, and 1 of them was involved with nitrogen transport, which is what happens during senescence, so this is pretty supportive of our hypothesis. What's shown here is the triple mutants having increased expression, shown in blue and green, of these nitrogen-related genes, while wild type, shown in red, stays low. So flowering time seems to be inducing expression of these nitrogen-related genes, while they're staying low in the vegetative wild type. But we were looking for more signaling molecules that could be doing this, so we compiled a list of all the differentially expressed genes from this analysis. There was 122 total, shown along here on this axis. So each lane across here is a different gene. And then every single wild type sample is down here in the corner. So when wild type has low expression, the triple mutants have high expression, shown here in yellow. And inversely, when the wild type has really high expression, the triple mutants have low expression, shown in blue. So there's like an inverse relationship between their expression values. From this list of 122 genes, I looked for gene expression changes occurring with genes that could be regulative meaning a gene that can actually change the expression of a different gene that's causing sort of a signaling pathway that could be signaling the onset of leaf senescence from flowering time. So from this, I compiled a list of 20 genes that seemed like they could have that capacity. I then looked at publicly available data sets to confirm these gene expression changes in wild-type tissues, meaning that they do in fact occur with flowering time in wild-type plants, not just the mutants. And then from this list of 17, I isolated 17 null mutants of these and then screened for uncoupled phenotypes. So uncoupled, meaning that the timing between bolting and senescence was different than what was seen in wild type. So after studying these 17 mutants, I used a clustering analysis to ask which is actually uncoupled. Which one of these mutants flowers and senesces at a different time compared to wild type, or the time in between them was different. So the very first cluster here from K-means analysis is showing the wild type in mutants at bolting time, meaning they were all developmentally similar in terms of their chlorophyll phenotypes and their gene expression phenotypes. Then the next cluster here is showing senescent tissues, also clustering with the chlorophyll and gene expression phenotypes. So there's a cluster of all the tissues at bolting. There's a cluster of a mixture of the mutant and the wild type 12 days later during senescence. But there's a third cluster of plants, of tissues I should say, that don't cluster with the rest because they are not senescing at the same rate as all the other plants. So what that means is there's a normal rate of senescence occurring between these two clusters, a mixture of wild type and mutants, but there's a third cluster here that does not senesce at the same rate, so we're calling those uncoupled. And an example of that, one of those mutants is XBAT35. So XBAT35 flowers at the same time as wild type. It has the same weight at bolting shown down here, and the same weight during senescence, so we can say that it's growing the same. But over these 12 days, wild type has a significant decrease in chlorophyll, while XBAT35 does not. And then wild type has a significant increase of this gene expression of NIT2, while XBAT35 does not. And if you remember, NIT2 is one of those genes that has high expression during senescence. So this together shows that XBAT35 is not senescing at the same rate as wild type after flowering time, which is supportive of our hypothesis. XBAT35 is an E3 ubiquitin ligase, so it adds ubiquitin to a group that marks it for degradation, and it interacts with something called accelerated cell death 11 to induce cell death. So cell death is an important component of senescence in the aging dying process, so this is kind of exciting to see because XBAT35 appears to be changing expression at flowering time to induce cell death, which is an important component of senescence. Here's data from another lab showing that XBAT35 is highly expressed in senescent leaves, but not as highly expressed in vegetative rosette leaves over here. So that was cool to see our data sort of confirmed in other growth conditions. Future steps involve measuring the expression of other senescence regulators in XBAT35, including, I also want to measure the expression of those nitrogen genes that I mentioned earlier in this mutant to see if they're downstream of XBAT35. I also want to measure another regulatory, like a regulatory hub of expression genes from XBAT35, and this will allow us to define where XBAT35 is operating in the pathway that regulates senescence. I also want to measure the seed nitrogen content to see if this delay in senescence that we see in XBAT35 translates over to a change in the seed nitrogen content that we would expect to see in agriculture. All of this coming together, we hope that we could classify this as a mutant that could be used in agriculture, that if there are stress conditions and a crop flowers early, if this mutation is present, there's possibility it may not senesce early, and it may be able to optimize the nitrogen mobilization process during stress in agriculture. So with that, I would like to thank the NIH for funding this, Dr. Bruston and the Bruston Lab, and then Kay Kamenesh for helping me